RNA-seq to study sexual reversed Gulf pipefish. Andrea.
Thank you. This is my master's work that I'm finishing over the summer, actually, and in the fall I have plans to start my PhD over at Florida International University. And so I'd like to start by explaining what is sex bias. Sex bias is when one sex transcribes or expresses more of a gene than the other sex. Sex bias can be sex-specific, where only one of the sexes even expresses the gene, or it can be male or female bias, meaning that both sexes are expressing the gene, but one more than the other. And sex bias is one of the mechanisms by which we get our fantastic phenotypic differences between males and females. And sex bias also varies by tissue type. So for example, in Sharma et al., they studied the guppy, which I've pictured here, and they looked at brain, gonads, and tail. And what they found was the most sex-biased gene expression was found in the gonads, followed by the brain, and then the tail. And although the gonads make sense, male and female genitalia are very different, the tail also returned a significant amount of sex-biased gene expression. And so why would the Gulf pipefish be a good candidate for sex-biased gene expression? For one, they belong to a family of fishes known as Syngnathidae, which includes the sea dragons, seahorses, and of course, pipefish. And a unique life history trait of Syngnathidae is male brooding, and it's shown in various forms. The Gulf pipefish has a very thin brood pouch, and you can kind of see the orange eggs showing through his pouch. Another interesting life history trait of Syngnathidae is the wide range of sex roles that they may exhibit. The Gulf pipefish is specifically a sex role reverse species, meaning that females compete for mates, and because of this, females tend to exhibit more secondary sexual characteristics, such as pigmentation and size. Down here, this is a male Gulf pipefish and a female, and you can see the female has brilliant stripes going down her body, and her body is significantly larger. This trait is part of what makes my study so unique, in that so few studies have even began to touch on sex bias and gene expression for sex role reverse species. And so in traditional sex role studies, we see that males tend to have more sex bias genes upregulated than females. And because of this observation, it's been hypothesized that evolution of sex bias genes is being driven by sexual selection. So initially I'm thinking, will my females have more sex bias genes upregulated than males? In addition, I was wondering if there would be sex bias gene expression in the brain and gills. Let me tell you why I've chosen these tissues. For the brain, I took the whole entire brain. When I opened up the cranial cavity of these tiny little fish in the field, there was no way to decipher what parts I was going to take, so I took everything. That being said, the brain is the main control center for most functions in the body. And the hypothalamus pituitary interrenal and hypothalamus pituitary gonadal axis controls sex steroid biosynthesis. So the brain tissue is a central organ in the sexual dimorphisms that we see between males and females. My other tissue is the gills. And the gills are a mucous membrane that are open to the environment, the first to encounter pathogens. I chose this because there are known differences between males and females in their immunocompetence, and I'll go into more detail on that later. So in addition to these questions, I wanted to know, well, if there are sex-biased genes, what genes are they? And then are there different functional pathways between males and females with these enriched genes? So as you can see, my research is very exploratory. I'm just reaching out there to see what could be out there for a sex role reversed species. All of my samples came from Port Lavaca Bay, which I have pictured here, and I was able to obtain 4 males, 3 females, just enough for statistics. So we employed Illumina paired-end sequencing to build the first transcriptome of the brain and gills for this species, and we annotated it using the Swiss Prot and Zebrafish database. We then did statistical analysis for gene expression, sex bias and gene expression using HR, and we got our gene ontology terms from Blastio. Our transcriptome came out really nicely, and after layers and layers of filtering, I was left with a total of 81,361 unique contexts to deal with. And we decided to use a combined data set of Swiss prot and zebrafish annotated genes for our DG analysis, or differentially expressed genes analysis. We came out with a total of 90, 73 in the brain, 29 in the gill, and 12 of them being upregulated in both brain and gill. Here's my breakdown of the DGs. On the y-axis, here I have number of genes, and then brain, female, male, gill, female, male. Right off the bat, you see that males have more sex-biased genes upregulated than females. And there are not that many sex-specific genes. Gene ontology terms, I have a few at the bottom here, can be really helpful when you're trying to decide what this gene might be operating in, what function is it working with. But when you look at them alone, they don't always make a whole lot of sense. So I created functional categories that give us kind of a big picture view of what these genes are operating in. And I found that there are genes being upregulated in functions that aren't upregulated in males, and the same for males versus females. A few for the females is tumor suppression and epigenetic modification, two of which there are no genes upregulated for males. And then in males, there's oxidative stress response, neuronal development, and transportation. And so you see the same pattern in the gills. There's several functions upregulated for females and not for males. But I want to call your attention to the immunity pathway. Both females and males have several genes upregulated in immunity. However, males have a few more than females. Anyone who's worked with next-generation sequencing knows how much data you get back from this method. And so for the sake of time, I've chosen just a few topics that pertain to sex bias and gene expression, of which I found differentially expressed genes. The first is sex steroids in the brain. And so I found 6 total that pertain to this topic, 1 upregulated in females and 5 in males. And only 1 really stood out to me and made a whole lot of sense, and that's testosterone 17-beta dehydrogenase 3. It catalyzes this reaction to create testosterone. And although testosterone isn't normally an active sex steroid for fishes, it is the precursor for 11-ketotestosterone, which is an active sex steroid for male fishes. So it's more of what I didn't find that's really interesting here. These 4 genes are normally found to be sex-biased in other sex-biased studies, and I found all 4 of these in my data set. They were annotated. However, none were differentially expressed. This raises questions about these pipefish. Are they operating different than other fish, or even other teleos fish to be specific? Or is this a sampling bias for the time that I took my samples? And the other topic I want to talk about is immunity. So time and time again, we see females have better immune systems and live longer lives than their male counterparts. And it's been thought that male and female sex steroids are mediating this difference we see. However, there's been discrepancies as studies went on to look at this hypothesis. In 2011, Roth et al. Looked at a sex role reverse species, Signathus typhoid, and he found that his males had a more active immune system than the females. And so this other hypothesis is out there that life history traits, rather than the sex, is really mediating this difference. And what I found is my male gulf pipefish in both the brain and gill had more immunity genes, or genes related to immunity, upregulated than females. And when I looked into them, more of them were attached to adaptive immunity. So in conclusion, I was able to answer my exploratory questions, including that I found males had more sex-biased genes than females. However, does this really mean that sexual selection is not driving this difference in sex-biased genes between males and females? Could it be a sampling bias? Would I see the same thing in gonads? Sampling time, season development, mental time might make a difference, or even my sampling size might make a difference. Either way, I've generated a lot of new exciting questions for us to go out and explore for the Gulf pipefish. And with that, I'd like to thank Courtney Bell for the help in the lab, UT Southwestern Center for doing our next generation sequencing, and TCU for the financial aid to come speak with y'all today. With that, any questions? Yes?
So maybe titers of some of these hormones may not be different amongst your two sexes. Maybe you don't see differences in enzyme expression. Maybe the receptors, for instance.
Yeah, that's actually what I looked for, and those were the receptors that I found, but none were differentially expressed.
And just one more question. So does the pipefish phylogeny provide opportunities for you to use a more comparative framework? So for instance, other species where there's this reverse sexual dimorphisms that are less or absent, more traditional, so that you could make these comparisons? And better understand the developmental genetic basis of sexual dimorphism. So could you compare this highly reverse sexual species where sexual dimorphism is more traditional?
Oh, yeah, definitely. So like in birds, that might be a good comparison. There haven't been a lot of studies on sex role reversal in birds. However, the males still hold that pretty phenotype, you know, where they're more secondary sexual.
Pipefish, do they present that opportunity?
In pipefish, no, for the most part, if they are, if they're not sex role reverse, they tend to look more similar. They don't have the. Yes.
Is there another hypothesis for why there are more male bias in genes?
Yes, there are a few out there. One is that maybe it's just selection, the fitness in the environment in general is actually mediating it. I haven't looked into that a whole bunch. And then there was another one. I actually kind of like the fitness in the environment, but there was another one. I'm sorry, I can't think of it right now, but I can look it up and we can talk about it. Yes?
So were your males pregnant or not pregnant?
They were pregnant. All my males were pregnant, and one of my females I caught actually spit out eggs, so they were inbreeding.
Well, that's the end of the talks for this session. So